In a small laboratory in the 1980s, Oliver Smithies made a discovery that would forever change how we understand and manipulate genes. His pioneering techniques in gene targeting would eventually lead to revolutionary treatments for diseases once thought incurable. Born in Halifax, England in 1925, Smithies showed early signs of scientific curiosity. As a child, he built his own telescopes and radios, foreshadowing a lifetime of innovation. After earning his doctorate from Oxford University, he moved to the United States, where his most groundbreaking work would take place. The path to scientific revolution is rarely straight. Smithies initially worked on protein chemistry, developing gel electrophoresis techniques that scientists still use today. This method separates proteins by size and charge, allowing researchers to analyze complex biological samples. But his most profound contribution was yet to come. In the 1980s, Smithies perfected a technique called homologous recombination. This process allows scientists to target and modify specific genes with remarkable precision. Before his innovation, genetic modifications were largely random affairs with unpredictable results. Smithies changed that by developing methods to replace or alter specific genes in living cells. The implications were enormous. Suddenly, scientists could create animal models with precise genetic modifications that mimicked human diseases. These transgenic animals, particularly mice, became invaluable tools for understanding conditions like cystic fibrosis, hypertension, and various cancers. What made Smithies' approach revolutionary was its precision. Rather than adding genes randomly to an organism's genome, his technique allowed for targeted modifications. Scientists could now delete specific genes, replace them with altered versions, or introduce entirely new genetic material at predetermined locations. This breakthrough laid the foundation for what we now call gene therapy. The ability to correct genetic defects at their source opened new possibilities for treating inherited diseases. Today, patients with certain genetic conditions can receive treatments that directly address the underlying genetic cause, rather than merely managing symptoms. Smithies' work also accelerated the development of knockout mice, laboratory mice with specific genes disabled. These models help scientists understand gene function by observing what happens when particular genes are missing. In 2007, Smithies shared the Nobel Prize in Physiology or Medicine with Mario Capecchi and Martin Evans for their discoveries concerning gene modifications using embryonic stem cells. This recognition came after decades of persistent work, often in the face of skepticism from the scientific community. Perhaps most remarkable about Smithies was his lifelong dedication to science. He continued active research well into his 90s, maintaining the same curiosity and work ethic that characterized his early career. His laboratory notebooks, spanning over 70 years of research, reveal a methodical mind constantly pushing boundaries. The techniques Smithies developed have become fundamental tools in modern molecular biology. Every time scientists create genetically modified organisms to study disease, they build upon his innovations. Every gene therapy treatment developed owes a debt to his pioneering work. Oliver Smithies passed away in 2017 at the age of 91, but his legacy lives on in laboratories worldwide. His work reminds us that scientific breakthroughs often come from persistent curiosity and methodical experimentation rather than sudden inspiration. The precision medicine revolution we're experiencing today, where treatments are tailored to individual genetic profiles, stands on the foundation Smithies built. His innovations in gene targeting transformed how we understand, study, and treat genetic diseases, saving countless lives and improving many more.